So good evening, everyone, and a very warm welcome to the CAPS Talk series. So we at CAPS are very proud and to share it with you that we are celebrating our 25 years at NCDS, and the team is led by Professor R. Sadamani, and then she will take you through a journey of the CAPS and talk about different themes which were covered in the lab. So over to you, ma'am.
Thank you, Shaila. Good evening, everyone. So happy to present some of our work today. Okay, so you can see the screen, and what we have here are going to be a series of talks, as Shaila mentioned. So to begin with, we want to today share some stories on our sequence search journeys, that is searching for protein sequences. And to just give you the introduction or to lay the story in front and the context, there has always been fewer number of protein structures and far more numbers of protein sequences. And this is because even though the technical advances mean that we can have more protein structures and there are initiatives for them, this requires a lot of hard work, and some of them are just not amenable for structural determination. And several of them happen to be complex proteins, and therefore we have always had fewer protein structures and more protein sequences. As if this is not enough, what is happening is that due to the post-genome sequencing era, as and when many more genomes got sequenced, the sequences, genomes of model organisms got known, we seem to be knowing a lot about mere protein sequence information, but we do not know how do they look like and what sort of function do they perform. So this is how it has been, and you can see this chart on the right-hand side shows the numbers. In front of us, they're all in millions, and you can see that in proportion to the numbers of proteins whose structures are available, which is 116K, you have a large number of proteins and 249M sequences. And the rate at which these kind of pouring into public domain databases, such as the names mentioned here, suggest that the rates are also quite different. The number of structures that get deposited the rate is small, and in comparison to the number of protein sequences that get deposited, the rate is much, much higher. So how can computational algorithms help with this huge burden? How can it help to reduce the kind of distance or the gap between the number of protein structures and the sequences? And for this, today, we are going to present to you 3 stories. The first story is going to be on using motifs of proteins, and the second story is going to be on using cascaded sequence searches, so namely repetitive sequence searches. And lastly, we are going to also explain to you as the last story how we have tried to compile this information and try to bridge the gap, that is, how many proteins sequences can reliably be connected to the protein structures using computational algorithms. So the first story goes as follows. A minute, I should just go to the next slide, which will happen. So it seems nature is just waiting for us to appreciate this huge situation in front of us. Okay, as we are carrying this message across in our mind, there were quite a few thousands of proteins whose structures are available, right? And there are structures of all types, and these are what are called as a forms. And out of this, let us imagine for a moment the smallest protein fold, a little fold, but it's also pretty popular. It is called interleukin, and it is a very important protein, but it's a small one. So let us take this interleukin 8 as an example, and you can see that if you put it up in a color coding such that the N terminus is shown in blue color, and if it's in a rainbow representation, by the time you reach the C terminus, it's going to be in red. All we can see by tracing this sort of a cartoon is that there are 1, 2, 3 beta strands, one follows the other, and then it ends with the alpha helix shown as a spiral. So each of these arrows represents beta strands. So why are we now talking about this structure of this little protein interleukin is because this is a domain. And quite often, protein genes can have multiple domains, not only one domain like this, but many domains. And within each domain, you have actually chemical groups that are arranged across like a string. Every amino acid contains a chemical group in its side chain, and therefore every time two amino acids come together, what nature is doing is to presenting and letting these chemical groups coexist in space. And when we do this, some of the amino acids have to remain where they are, and some of them are allowed to change over the evolutionary time scale as nature would like to do. And nature will not like to compensate on function because structure has to be preserved. It cannot just open up and become denatured. And the protein also has to preserve its function. So I guess what we are asking is, out of the millions of protein sequences that are coming through these genome sequencing projects, if you were to ask a question, how many of those millions of sequences are likely to adopt this little interleukin fold? That is a simple question. And for this, we took an approach which is quite different and complementary to the other approaches that were floating around in other bioinformatics labs. Most bioinformatics labs were worrying about overall sequence identity. That is, if you are imagining millions of protein sequences, which of them are closer in sequence to this interleukin, and that might give a clue about how many other proteins can adopt this fold. And that means the structure sequence space is getting bridged now. Whereas few others were concentrating on what I call as the functional groups of proteins. That is where the business end of the protein will reside. What we did in our lab is to attack this from a completely complementary angle, wherein we asked, out of these several residues that are out there with their own chemical groups in their side chains, which one of them are important, not for the function, not driven by sequence identity perhaps, but what is important for the integrity of the fold? What keeps the fold remain that way? Namely, these are structural motifs. And when we asked this question, computationally, we decided to go through a stepwise procedure. So I'm going to tell you in this story, there are 3 steps. The first step is going to gather all those proteins which we already know, we believe that they belong to the interleukin family. And then we go and search in that sequence database for only those members, and then find out their relatives, and then align them and find out where are the amino acid changes going on. The chemical groups, are they changing very much? And which are the places where they don't change? So what we have in the x-axis are the residue or the amino acid numbers, and what we have in the y-axis will show how much residues are changing when we gather other interleukins from the sequence database. And you can see that some places just cannot tolerate substitutions. They remain highly conserved, but some other places it doesn't seem to care. Many amino acids are changing, so this is an information. In informatics, we always look for patterns, right? So this is one of the patterns. So the second step that we decided to do was to then ask, out of these many regions in the protein, in the interleukin, as in this example, that remain conserved over an evolutionary scale, how many of them are talking to each other in space? Because for instance, you have a research institution or an academic institution, you may have 10,000 students, 1,000 students. Not all of you will be making all decisions, right? There will be a dean, there will be a director, there will be a head of the department who will sit together once in a while and make important decisions. So also here we are asking, out of the many conserved regions, can we find out which of the conserved regions shown in green color here are they really talking to each other? Spatially proximate, as the name is called. Space in 3-dimensional space, proximate as in close distance. And we can keep on measuring these distances because after all, we have a structure in front of us. And then we select out only those regions, and these conserved regions are now called as motifs or structural motifs because they are beginning to be so important that they are always talking to each other. So maybe they are important for the integrity. So these structural motifs are mapped on the structure here and on the alignment down below. And in this example of interleukin, we have just taken 2 entries. That means for both entries, we go and look for the homologous sequences. And that's the third step, which is then ask the question, out of the 2 members that we have their own structural motifs, how equivalent are they? And we can see when we put them side by side, the brown strand and the brown strand here, they are remaining as not only conserved motifs, but also structural motifs because they were in spatially interacting distance with the blue strand shown. So the brown strand and the blue strand are structural motifs. So this is an important step. And how can we use this in sequence searches? How can you use to bridge the gap between the huge expanse of protein sequences and the small number of protein structures? For this, we use these structural motifs as constraints and go and search in different genomes. To begin with, when this story started around 2001, we had just got our Wellcome Trust Fellowship, in which the main component of my Wellcome Trust proposal was to look for structural motifs, use them as constraints, and search in genome databases. And when we tried it around 100 genomes, the good news we got was our interactive motif-driven sequence searches are much more powerful in comparison to a very popular and highly used sequence search algorithm called SciBlast. SciBlast itself is pretty sensitive, and you can see the number of relatives that the interactive motif constraint search is bringing up is much more. And this happens for a wide variety of folds, so that way we became quite happy that the structural motifs are indeed quite important and could be used as primary drivers or constraints to then focus our sequence searches, and the sensitivity also seems to be pretty high. And you can also do something more with the structural motifs, which is to use them as sole and simultaneous constraints. And now, unlike the SciBlast or even the pattern-assisted SciBlast, where we care for the entire protein and your similarities, we are only focusing on the different motifs. And this is now being written up by one of my first PhD student, Saikal Chakravarti, and he he wrote an algorithm called ScanMod, by which you can use these motifs as sole and simultaneous constraints. And interestingly, we are able to pick up many more proteins who were never annotated, means nothing was known about their function, and they are there coming from a genome database, and we are able to now recognize them simply because we know these are structural motifs which can guide the strength of that particular fold, and they are present in these sequences. So that seems to be sufficient evidence to recognize them as putative members. So this is a part of the story. But in reality, what happens is we are not left with a simple small protein like interleukin. In reality, you can have protein domains which are much more complex. Why? Because either you can have not two members in the superfamily, but many more members in that superfamily, but you can have many superfamilies because you have just taken one interleukin one. But also some of these structural folds could be pretty complex because they could have multiple domains, they could have insertion domains, and they could have what are called as discontinuous domains. And these are kind of intuitive terms because insertion domains means something is getting inserted in between, so things become very hard to search and find. And then discontinuous domains means in the sequence, it could be going up and going from one domain to another. So for all these reasons, it's not easy always to use CanMod or to use the principles of structural motifs as our lab had suggested. So for this, what we had done years ago, was to first of all appreciate that there could be multiple members in a superfamily such as this. This is the same interleukin example, not with two examples anymore, but many more as we can see of late. And each of them have been color-coded so that you can recognize the secondary structures. The beta strands, those which are shown in the blue color arrow in the cartoon, are shown in blue color, and the single helix that we saw at the end of the interleukin fold is shown in red color. So these are real examples whose structures are already known. This is to just show you the numbers are increasing with time, even for structural entries. And then you can have complex structures, right? This is a fold where if you try and overlay them, and in technical terms, it's called as a superposition. If you put one of these members at a time on the other, which happens for another superfamily, which is that of a methyltransferase enzyme, you can see that the overlap is not good. That means there are lots of structural variations that are happening. So it's going to be hard and harder to handle such protein superfamilies. But we have used these sort of techniques and already built a pre-curated database, which is called as IMODB, and it stands for interacting motifs that are contained in the form of a public domain free database, wherein the number of motifs have already been recorded for all the superfamilies, and they're also provided in an objective manner so that you can know not only how many motifs are there, but which are the two motifs that are interacting with each other most. So these are presented as pseudo energies. So that if the energies are low and negative, that means such motifs, such pairs of motifs, as shown in this matrix here, will be highly strongly interacting with each other in three-dimensional space. So the more negative it is, the more strongly these motifs are interacting. So one can just choose which motif to use as constraints in our sequence searches. So that is the first story on use of choice of structural motifs to guide sequence searches. I hope you found this pretty interesting, and the journey was also a very smooth one, but it went through a number of years before we landed up with the IMODB. And we are still continuing to update this database, looking at single member superfamilies and so on. So I don't have the time to go through because I want to tell you the next story, which is on cascaded sequence searches. The sequence searches, like in the SciBlast, which we were comparing with the motif-assisted sequence searches, can we use them again and again? This is the question that we asked. And the reason why we asked this question was supplemented by a sufficient amount of valid literature. These papers are shown down below. This were coming up in the late 90s and early 2000s, and the message that comes out is very, very simple, but very powerful. For instance, if you have a protein family such as interleukin, which let us call that as family A, and then you have another protein family, say in interleukin X, which is very far, many sequence changes have happened. So it's not going to be easy for SciBlast to connect any members of family interleukin H to any members of this family interleukin X. Let's imagine that's the situation. You do a SciBlast, or even the structural motif assisted SciBlast, you get nowhere. You don't get to see family B in your hits. Then what happens is the message that came from these papers, which said, instead of trying to reach that family B, which in this example is interleukin X, don't be disheartened. Don't be disheartened, and instead what you might want to do is to take the help of the hits that come and use them as start points, and then do your sequence searches again. And these hits are called as intermediate sequences, and the name is very intuitive because they serve as intermediates to hopefully connect family A, interleukin 8, with family B, in this example, interleukin X. And they had shown, such groups have shown that already this logic is so powerful that you can increase the coverage and get much more members of the family B to be recognized by simple computer algorithms such as SciBlast or even motif-assisted SciBlast. Let's see how this can take us far in this story. And this is a story that, like I said, started in the early 2000s. And the pioneering papers that I talked about the intermediate sequences searches were led by one Professor Cyrus Chotia in Laboratory of Molecular Biology, Cambridge at that time. And he was the one who proposed intermediate sequences could be powerful. And along with my collaborator, Professor N. Srinivasan, in Indian Institute of Science, we together, both labs, my lab and Srinivasan's lab, took it forward and asked the question, can this help us to bridge the gap between the huge amount of protein sequences and the small number of protein structures that we have? And for this, we would conceptually have started with family A and did one round of SciBlast and gather homologs and relatives and then you call them as hits. And the fun doesn't stop there, because you take each of these relatives as start points and you repeat your SciBlast, and this is now going into what we call as the second generation sequence search. And then you gather new homologs and use them as start points, and they go over to your search, and then you get few more homologs until you reach the family B. So it's very fascinating to imagine simple sequence searches by using these sequence. We are literally have Professor Cyrus Totia alive, nor Professor N. Srinivasan alive. We lost them rather recently. But whatever they have carried out on the sequence search arena are a wonderful schema. And I'm just going to take you through the next few minutes on this particular story. All right. So these are going to be, as you can So are you able to see the slide on sequence space? Okay, so for this, let us imagine as though your hello, I think you're not able to see the screen. I'll just share again because we got out of Zoom for a minute, but just let's try to connect back onto Zoom. Just give me a minute, please. Okay, share screen, go back, and I will go to this one, and then full screen mode. Got it. Yeah, it's fine. Okay. So now let us imagine your that this whole sequence space. It's like a starlit sky. So you have family A somewhere here, and you have family B somewhere there. Depending on how far they are away from each other in the sequence space, and how much they are able to talk to each other, you can sometimes, or you cannot sometimes use computational sequence searches to make these connections. That's why intermediate sequences become powerful. And that's what we discussed, Cyrus Jyotia's proposal, and our collaboration is leading towards. And for this, we have to take this objectively because we discussed every generation of your sequence search, keep them stringent so that you're not walking into wrong parts of the sequence space. And even as this is going on, how exactly is it helping? And for this, we can appreciate it better by means of recognizing every member and then projecting them in the form of a tree diagram. And here, those sequences, protein sequences that are close to each other, there's close relatives will come clustering together, and those which are far away from each other, such as the family A and family B, are going to be far away and appear as different clusters. And as and when we keep doing this different generations of sequence searches, and the generation 1 gave rise to hits, but it was not enough to connect sequence the family A and B. And the second generation shown in red color were the ones that are literally able to fill the sequence space and be bridges so that the family A and family B are beginning to see each other better. And in doing so, you can imagine, and it happens that the third generation shown in green color are those which are actually able to make that important connection so that two different families, A and B, are able to get connected. So this is conceptually how a cascaded sequence search can work. However, there are instances where we know that two protein families A and B are connected. You do the cascaded sequence searches, you take them through multiple generations, generation 1, generation 2, generation 3. You keep doing these searches and still they refuse to get connected. But we know that from biological function characterization that indeed these two protein families are related. So are we going to give up here and say enough? We can neither use cascaded cyblast, we cannot use structural motif-driven cyblast, or you cannot rely on cyblast anymore. No, but for this we can do something else, which is to use computational algorithms to our advantage and to imagine to just literally generate artificial sequences using computers. And when we do this sort of artificial sequences, you design them such that it's not a crazy, chaotic design, but an intelligent design. And how does this knowledge come from? We look at the known protein structure, proteins relatives, and then we know certain points. Some amino acids are getting tolerated, some other points, amino acids are just anything. And that we had seen in the step 1 of our interacting mode, structural motifs as well, right? So we use that information and then project that knowledge in the form of a pie chart. And it is now like a kind of a roulette wheel. And then you assign a random number and then let it hit at any position of your protein, anywhere in this pie chart. And the pie chart is not random, it is derived from the knowledge. So that random number is going to hit on a place which is biologically meaningful. In the sense, only amino acids that have similar chemical groups are going to get exchanged. And when you then create an artificial sequences, it has a biological meaning still. So we're not moved too far enough that we cannot control this generation. And when we have these sort of artificial sequences, it helps to bridge the gap so that family A and B are at last getting connected. And all those shown here on the phylogenetic, on the tree diagram on the right hand side, the ones shown in light blue color are the ones that have been generated in the computer. These are random sequences, but not too random that they are drifted away from either family or A or B. And those members that are belonging to family A and B are shown in golden yellow color. And you can see how meaningfully and beautifully these artificial sequences are getting populating themselves all around this family A and B, so that this computational algorithms can now begin to pick up and do better with respect to their coverage. We are still not solved the problem, and I'll tell you why. Because sometimes you know that family A is isolated from family B. After all, your random sequences are making sure that they are not totally chaotic and stops to make sense. However, family A has all the freedom to go and get connected to family C using these artificial sequences. How on earth are we going to control and make sure that family A will get connected to family B and not to something else, some other family? And for this, we can actually use both family representatives, or what are called as mathematical models of both families A and B, and then generate your random sequence so that your artificial sequence is somewhere right between the two families. It looks a bit like family A and a bit like family B. And then these sort of artificial sequences are even better in making connections. That is what is shown here. So then you can use that sort of information back onto the general question about how good are computer algorithms. How can we train them? How can we develop them so that the sequence searches become more effective with high coverage, which means the sensitivity is high, but not compromising on the specificity so that it's going on to wrong families and poor connections onto drifting into the sequence space and so on. So these sort of cascaded sequence searches are very powerful and they are very exciting. They do not compromise on both sensitivity as well as specificity. But what are the bottlenecks? The bottlenecks are that they are computationally very demanding, right? When I talked about Professor Cyrus Chothia's suggestion, I had mentioned these are demanding on computational time. So it really depends on what computer server you're using. If you have a poor server, a relatively poor server, it's going to take hours and hours to make these computations, because at every generation you're committing to more and more sequence search runs, which are called as cyblast in this case. And then on the other hand, if you had a powerful computer cluster, things can be managed very fast. So you really have to know how to proceed. And this also going to the better understanding of how and when to use these sort of powerful sequence searches. And in this second story, I've had both Sandhya Sankaran and Richa Mudgal as key players, along with in Srinivasan and in the cascaded sequences. On the other hand, in the motifs story, which I said as the first one, we have had my very first PhD student, Saikat Chakravarti, who had used the structural motifs, and my second PhD student, Anirban Bahaduri, had been worrying about how to use the interacting motifs, and together with Pugalendi, Anirban and Pugalendi had generated the IModDB that I was talking about. They had also done wonderful other things, which will actually go on to the third story. How are Anirban and Pugalendi involved? This happened almost by chance. That's the third story, which is to knowing that these interacting motifs are powerful drivers to improve the coverage, as explained in the first story. How can we use it to actually bridge the gap between protein sequences and structures? After all, what we have seen earlier was only to apply it on a handful of protein folds and about 100 different genomes. But now, and even then, in 2004, when we started these sequence searches, there were many more genomes, many more protein superfamilies, domain superfamily. And for this, Anirban started off to use our weak-looking cluster. And at that time, we had, with the Wellcome Trust grant, had built up our very first cluster, which we called as Apollon. It's not an athlete, it's Apollon machines. There were only 11 dual processors. And in fact, Anirban used to call the cluster room as Siberia, because it was kept very cold, and every now and then we had to go there to make sure the computers don't get heated up. But this story is in the year 2004. Nowadays, we don't ever get to see our computer clusters, not only because we have efficient IT staff, but also because if the system gets heated up, there are automatic ways by which they will get switched off, so you don't have to go and babysit them so much. But that was also fun because he used it quite nicely, wisely, and he used all the Apollon machines and put them on a continuous series of sequence searches, and it went on for 3 full months. And then what we were left with is the next challenge, which it starts to occupy an awful amount of computer disk space. And we were finding ourselves with a very rare situation that all the lab computers in our lab then in 2004 were filled with the results that Anirban and Pugalendi were generating using this sort of Apollon. And what were they doing was to simply ask the question, can we now use the interacting motifs and few other sequence searches like SciBlast itself, and then apply it on all the superfamily, all the domains whose structures are available, and against all the genomes which we can lay hands on. And that means so many model organisms. So these are definitely very time consuming. That's why it took 3 months, but it is a one-time intense computing. And once it is done, you can work on 2 different hierarchies. You can work on a structural hierarchy. That means you can look at different protein forms and then look at protein forms and domains that have similar biological function, which is called as a superfamily. And you can also do one thing, just because you have now run it on many different model organisms and their genomes, you can work across the different taxonomic hierarchy. Isn't this beautiful? Because then you can ask simple general questions such as how many interleukins are there in the intermammalia, and lo and behold, these answers are immediately available because the results are pre-calculated and stored in this database that we called as GenDis. GenDis standing for genome distribution of protein sequences starting from proteins whose structures are available. And this was a very beautiful, fun exercise. Just because it went on well, we didn't stop there. Anything that goes well has to keep going, and that's when things became even more interesting, because as we went on, what happens in 2004 does not remain the same way in 2014 and so on. 10 years later, or even 8 years later, we find that not only you have better computer clusters, but importantly, your protein sequence database is growing, right? There are many more genomes whose whole genome information is available. So it becomes more and more challenging to connect and bridge the gap between protein structures and sequences. And therefore, our journey continued in this third story. And in the next few minutes, I'm going to just take you through. I won't take long because the key players are going to come and join us in this discussion very soon. Okay, so here we have many more sequences, and how more. And that was actually, you got a glimpse of it in the very first slide. And to begin to address the huge number of protein sequences and the huge number of protein structures that are piling in, not only we need better computers, clusters, but we have to be going with the stream and learn and be in touch with the literature and see how many more algorithms are there. How many more bioinformatics algorithms are there? The interacting motif was our own baby developing from our lab. SciBlast was the key sequence search algorithm, but the PhiBlast is nothing but the motif-assisted SciBlast. And we also started getting other computer algorithms such as context dependence blast, as well as JACAMO. Then the question became very clear. Are we going to use all these algorithms and sit and make a consensus about what each method is saying as relatives for each of the proteins whose structures are available? Or are we going to select one best method and stick to it? And if so, out of the many members whose structures are already available, like for instance, interleukins, we saw that already there are many entries, right? Are we going to take all of them as start points or only one of them? To address all these questions, Advait Joshi, who was in our lab then, decided to look at a small number of protein domains of diverse folds, some of them which are rich in helices, some of them which are rich in beta strands, and some of them which are mixed of both helices and strands. And even by taking just a handful of such examples, 12 different domain superfamilies or folds or forms, as we can call, he then applied each of these algorithms to understand which method to use, which of the members to be start points, and so on. And this is just to show you that it's important to stick and recognize what is called as a best representative sequence. And this term is again very intuitive. Out of the many members that you know whose structures are available, which is going to be acting as your representative, because you cannot afford to use all of them. That's the first news, because if you use all of them, we will be doing these sequence searches for too long. It might take not 3 months, but maybe even 1 year to compute all these searches. So we have to make some approximations. And that's what I wanted to say, that is to use best sequence as a representative. And you should also do what is called as the comparative analysis, by which you take the results from each of these methods. And this is what was painstakingly done by Advait. And he showed which method can be used and how to recognize best representatives and so on. I won't go into the details. And in this slide, I want to just convey one thing, which is the numbers are increasing. So you can see that there are close to 2,000 domain superfamilies, like the interleukin 8, which we discussed, like the methyltransferase. There are actually 2,000 superfamilies out there. And they all have to be now brought in front of the huge expanse of the sequence database. And therefore, we are talking about large number of searches, right? So with this, I'm going to really stop and by introducing this third story. So the first story, to just remind ourselves, was on motifs, using structural motifs as constraints in our sequence searches so that the coverage and the specificity, sensitivity are all achieved. And then we discussed the cascaded sequence searches, where you give each of the relatives that come out of one run to become start points, and you do generations of sequence searches, and that helps to connect fairly isolated families like a family A and family B in the sequence base to now get connected. And then the third story, which is on actually being able to bridge the gap between protein sequence database and the structural database, using these sort of techniques and generating a one-time calculated repository such as GenDis. And all of these makes use of computer algorithms alone. And most of them require only sequence information of the new proteins, that is the sequence site. You are using the structural information as much as possible to recognize the motifs and so on, but that is only to drive specificity. Yeah, so with this, I will call upon Shafi to quickly take you through some of the publications where all of these 3 stories have been described in detail. So you can go through them. You're welcome to also write to us and ask if you have any questions. And after Shafi has taken you through these publications, I'm going to next call upon 2 people who are in the lab, 2 players who had participated in the 3rd story, GenDis. And these are Advait Joshi and Meenakshi, who had actually then had the courage to escalate them to include all the 2000 odd superfamilies. So very soon, Shafi is going to come on the video and explain to you on the publications. And after that, I call upon Advait and Meenakshi to come and talk about their journey from their perspective on GenDis, the 3rd story. Please, Shafi.
Hello everyone. So these are few of the most cited publications from our lab related to what ma'am talked about today. So the first 2 publications it's regarding structural motifs in protein families. One is from Chakravarti et al. And one is from Kugalandi et al. So the first one is SMOS, it's a database, and another one is SMOTIF, it's a web server. And the third one, IMODB, is from Kugalandi et al. It's called IMODB. And it's published in Nucleic Acid Research. And following to that, we have multiple publications for cascade searches, which do remote homology detections. And this list includes one review also. It published in 2016. It's protein sequence design and its application, published in Current Opinion in Structural Biology. And at the end, we have 2 publications for GenDis. One is from Kugalandi et al. And one is very recent from Meenakshi Iyer. Yeah, that's all. Overview.
Thank you. I call upon Dr. Advait Joshi and Dr. Meenakshi Iyer to come up and say their own experience on this interesting journey on GenDis, which was one of the highly visited web pages from our lab. And I suppose it becomes even more meaningful if it is accompanied with some questions. So may I request Vikas Tiwari to also come on video, and I'm sure he has some questions to address so that both Advait and Meenakshi can address them. Vikas, please.
Hello. Hi there. So I had few questions regarding the GenDis. So the first one is to how can you explain exactly the relationship between the increase in the sequence space and the structure space?
Thank you, Vikas, for the question. So as in the first slide, we have seen that starting with the structure space, so we have very high number of sequences. Every year there are a lot of genome sequences being deposited. We have metagenome sequences, samples being sequenced as well. So we have a lot of sequences deposited in the sequence databases. And even though the number of structures being deposited in the structural databases is also increasing every year, but the rate is not as comparable to the rate of increase in sequence space. So we have a lot of sequences being deposited in the databases every year. So our aim is to bridge this gap between the sequence space and the structural space by using the information between the related families and superfamilies within the structure space, and using this information to identify the closely related sequences or homologs from the sequence space. So we are creating a projection of the structural space onto the sequence space using the information sequence and structural information.
Okay, one more question. if I have a like human protein, how this GenDis database will help me in exploring the different functions of my human protein?
It's a nice question, because perhaps I can take it. So basically, in GenDis database for a human protein, whichever human protein you select, so you can look at what other organisms also contain that protein domain. So through GenDis database, you will be able to navigate through all those taxonomic levels and look in other organisms as well. Along with that, if your interested protein domain is found along with several other domains, so which typically is called as domain architecture, so that as well can be studied. So you can get an idea about how the protein domain of your interest is combining with other structural domains. And through those domains, you can again further navigate their taxonomic distribution and their domain architecture distribution.
Okay, thank you. One last question. Now that you have experience with multiple sequence search methods, which method do you recommend? Like if I have to start searching for an envelope for my protein?
Thank you for the question, Lucas. I can take this question. So as you can see in the slide, so the sequence search methods have undergone a lot of evolution over the years. So starting from sequence alignment methods, which were used for sequence searches like BLAST and FASTA, like it worked very well for closely related homologs. Then we move on to the, as Sodhamini ma'am had already introduced about iterative sequence search methods and cascaded sequence search methods. So we have a few examples of iterative sequence search methods, where at the end of the sequence search, profiles were created from the homolog sequences which were obtained, and that profile was again used to search the sequence database in an iterative manner. So these methods, the next generation of sequence search methods, they have increased sensitivity. They can identify remote homologs, and they also maintain a good balance between computational time and the number of homologs that we obtain. And then we go on to the next generation, which is a Markov model-based alignment methods, and we have few examples of the sequence search methods as well. So these are very highly sensitive, and the algorithm at the end becomes very greedy. It tends to accumulate a lot of number of hits, and many of which turn out to be false positive, because it makes this cross family or cross superfamily connection starts to associate with the neighboring superfamily. So if we are focusing on a particular family of proteins or particular superfamily of proteins, so then we, from our experience, we would recommend going for the profile-based alignment methods, wherein it has a good sensitivity, and also with respect to computational time and the number of hits, there is a good balance.
Okay, thank you, Adar, and thank you, Meenakshi.
Thank you.
Thank you, Vikas.
Okay. I would request the participants to fill in a form. It will help us to know how much you have learned and enjoyed this talk. So yeah, all of you, it's a very simple form. Just I think it has a couple of questions, so if you can answer them, it will be great. Thank you. Any questions? If you have any questions, you can raise hand, or you can just come on the video so that we'll know that you're ready to chat with us. Okay, so even as we are waiting for questions, let me just say that this is the first in our seminar series, and we started with the oldest project in a way, because the sequence search and structural motifs began as our lab also began in '98, and as soon as I started the lab in '98, in 2000 I had got this Wellcome Trust grant, and where I had put up the structural motif as a key project. And we were also we also could get a computer cluster in which these GENDIS runs to be performed in Siberia, as we call it. But then what is interesting is there are more and more stories. And what we want to tell you this month of February is are going to be stories on the sequence front, and then we'll go on to structure front, and then we'll look at how using computer algorithms we can begin to learn on protein functions, and then it goes on in different themes. So definitely next week, Thursday, same time, 3 p.m., we will be meeting with yet another story. Safely stay tuned. And if you have any questions, don't feel that this is going to be naive or so. Please feel free to ask now. Or if not, you can always reach our lab. Then I'm just giving you my email address just in case, so that you can write to us. All right. So yeah, thank you also to the core team who has been helping with this seminar series, and this includes many people. But today we've had Shailya Verma, Mohammad Shafi, Advait Joshi, and Meenakshi Iyer participating and in this seminar talk today. Thanks. Thank you. And thanks to all the participants who have been hearing this so patiently. Thank you.